up we have Gordon Jong from Morey University of Newfoundland going over eDNA barcoding and marine conservation.
Thank you so much for the introduction. As Priscilla mentioned, my name is Gordon Jong. I'm a PhD candidate at Memorial University, and today I will be talking about some of the data that we've collected in marine conservation or marine protected areas. I'll use them interchangeably today, off the coast of Newfoundland and Labrador to help us inform sampling design and detecting changes in community composition. Okay, perfect. So, reaching 30 by 30 was already mentioned today, but another part of this is monitoring existing areas and areas that we will eventually implement. We need to be able to make sure that we're able to detect changes across space and time, and if these areas do change and then conservation areas are not meeting their objectives, scientists and managers may have to implement more of an adaptive management. So since eDNA is relatively understudied in ocean surveys or ocean environments, particularly in the deep sea, actually I was Googling last night on Web of Science and I wanted to kind of figure out how many studies involve the deep sea, and it was less than 30. So it's really this environment that's been understudied. So just to maximize sampling time and fiscal limitations, just for reference, it costs about $20,000 to $30,000 per day, and that's just ship time when we're out there, right? So it's really, really expensive for us to go out and collect samples in these remote environments. So we really need a solid sampling design when we're out there. So through all the eDNA samples I've collected from these protected areas, my goal is to use them as more of a case study to determine the efficiency and efficacy in detecting change across the Northwest Atlantic and within the areas themselves, across the various depths that we sampled, as well as gains and distances, and gain some insight into the factors influencing those changes. So using the results from species richness and changes in community composition, we then can have some idea in the sampling effort and design required to detect change, and hopefully this can aid managers and scientists when doing their own surveys in these marine protected areas. So in 2022 and 2023, we collected 120 paired samples from Funk Island Deep, which is here, Hawk Channel here, and in 2024, Northeast Newfoundland Shelf Slope. We collected pair samples at the surface, which was about 5 meters, and then at the bottom, which was about 5 to 10 meters from the bottom, in each of these areas. And just so you get an appreciation of scale, these two smaller ones are about 1.5 times the size of PEI, and the larger one, the Northeast Newfoundland Shelf Slope, is about the size of Nova Scotia. And we collected samples from 25 to 200 nautical miles from shore, and the depth ranges from about 120 meters to over 2,500 meters, so 2.5 kilometers. And just for reference, 2.5 kilometers is about 5 CN towers stacked on top of each other, just so for a sense of scale. It's really deep. You should ask me after how we did it and how long it took to do it. And just so for a basic methodology, I don't really want to get into this too much, we use a CTD rosette to collect our samples. We transferred 4 liters of water into a sterile container. We then pumped that, we then pumped that through a pump and then shoved those filters in a -80 freezer while we're at sea. And then once back on shore, we extracted the DNA and sequenced using the CO1, 18S, and 12S gene regions. And then thanks to DFO, IML for the bioinformatic pipeline, Audrey and Genevieve, thank you for helping me navigate bioinformatics. We used that, and then this is what we found using those 3 markers. So after cleaning, and then from our assigned taxa, we had over 8 million reads from 352 species, but I don't really want to get into that today, and I mean, this is described by this basic phylogenetic tree here. What I really want to talk about is species richness between the different areas and the community composition changes between them. And so we can see through these species accumulation curves and extrapolations, we obviously didn't reach total species richness. We never will, not in the ocean. We had a lot of unassigned ASVs. But we can start to see some differences where our bottom samples, these three, were more rich than our surface samples right here. And that our shallower MPAs were more rich than our deeper one, the green one, northeastern Finland shelf slope. But we can also kind of see as we increase hill numbers or Q, the species richness, or the high species richness we're observing is due to unique species rather than shared species between the areas. But richness was also affected by the depth. So even though our bottom samples were more rich than our surface samples, as depth increased, so did richness. And in the Northwest Atlantic, this is contrary to the existing literature. So I was really excited about this, but some DFO managers, not so much, but that's okay. And this is just likely due to our multi-marker approach, which kind of casts this much larger net than traditional samples that the existing literature uses. And so we can kind of see some of the differences across bioregion, and obviously surface and bottom. We're not expected to find benthic organisms, coral sponges, etc. at the surface, but we can also start to see some differences between the northeast Newfoundland shelf slope and the two protected areas that are on the continental shelf, where we start to get some differences in our multivariate analysis using Jaccard dissimilarity. But also, I kind of wanted to look across how these species are changing across space and depth. So this is an inverse Sorensen dissimilarity, now similarity, GLM, just to wrap your head around it makes it a little easier, at least it makes it easier for me, across the function of distance. And this is for all the samples that we collected from each of the different protected areas. And we can see that as distance increases, and as we would expect, the communities changes. Now, this is a weak correlation, but as working in the ocean, we often have weak correlations because there are so many factors at play. And then we used a Spearman correlation from a Mantell test. Kind of wish I went first out of last presentation to determine significance, and they were significant, but that's because we had a lot of samples. Okay. Oh my goodness, there was a delay. And these relationships maintain true within each of the protected areas, where as space increased within each area, so did the communities, or they became less similar across space, except for Funk Island bottom samples where there was a lot of dispersion. I kind of want you to pay attention to the dispersion that's happening in all these plots, because it's going to be important when we talk about sampling design and sampling effort later on. And then looking through the depth of lens, so the difference in total water depth between the samples that we took, obviously there wasn't as big a difference in surface, but there was a stronger slope, approximately 5 times greater than the surface, when comparing communities across the bathymetric scales that we sampled. So that was again 120 to about 2.5 kilometers. And this maintained true within each of the different protected areas that we sampled, but what was really interesting to me is that at Funk Island, which had the lowest difference in depth, it was the shallowest one of them all, had the greatest correlation between depth and community, decrease in community similarity. So the beta diversity was really high across each of these protected areas where there was very few shared communities between sites. And turnover, not nestedness, dominated at both regions, or at all regions and both depths. So meaning that the assemblages differed by species replacement and they weren't being driven by nested subsets. So really the spatial structure is being driven by distinct local communities or distinct DNA, not richness gradients. And then we can also see how taxonomic communities were changing across depth for each of the markers, and this is just relative proportion of breeds, where we have a lot of heterogeneity in our shallower depths. I'm only going to really talk about bottom because surface is kind of boring. But we have a lot of annelids and anthropods at our surface, and then as we get deeper, we get a lot of Nidaria. And then looking at the taxa, it was predominantly dominated by jellyfish and not corals, as we would have hoped, because a lot of these protected areas are designed for corals. Now, I'm not going to go through all the species driving the differences in each of the areas. That would be really boring. But just to the species that are driving the differences make ecological sense. At the surface, we're getting pelagic zooplankton and marine mammals. At the bottom, we're getting seafloor benthic fauna. But interestingly, in Funk Island, capelin was a big driver. We actually hypothesized that this is an important spawning area for capelin, which is an important forage fish for many of the species in the Northwest Atlantic. Hawk Channel lanternfish and Arctic copepod. It was our most northern marine protected area, so it's going to get more Arctic species. And Northeast Newfoundland shelf slope, a lot of deep sea specialists. It's on the slope. It has a really big depth gradient from about 500 to, I think, about 4,000 meters. I also did a generalized dissimilarity model, and I'm not going to really go into it, but I'm just going to talk about the main results. How water mass, so the combination of temperature and salinity, were having the largest effect on community turnover. So it had a large effect for all when we cooled surface and bottom together, but it still had a significant effect for bottom. Space was also there, but not as noticeable except for bottom. So we can kind of start to get this idea that our bottom communities are being driven by water mass while being spatially distinct. But also, I kind of want you to note that the total variation the total explained variation is quite low. And then that's just kind of the downside of working in marine environments. It's this complex, three-dimensional environment where there's so many factors going on. You have currents, you have temperature, salinity, habitat variation, patchiness. It makes it really hard when you're analyzing your data, by the way. So when I was trying to figure out what the sampling necessary and needed, my really focus was on the variation or heterogeneity and dispersion that we're seeing across space and depth and through our factors that we analyzed. So sampling depth, and not so much region, because it was relatively similar for all the regions, the heterogeneity, is probably going to drive our sampling effort needed. And this is true when we do a weighted allocation following, following stratum richness and dispersion, we are able to capture a lot more richness with fewer samples. So if you wanted to reach 80% of the regional richness, we would need approximately 75 samples compared to an even spatially sampling design, which would need 110. So you're almost saving like 1/3 of your costs, which is really important to consider when you're monitoring these massive protected areas. And this is the same for looking across change, collecting samples before and after. And where our surface samples, we need a lot less samples before and after a community shift at 5%, 10%, and 20%. Bottom samples is almost, we would need a lot of samples to determine small changes in community composition just because of the heterogeneity and dispersion that we are observing over time, but we would only need a modest amount of samples to detect larger changes, such as 20%. So things to kind of consider when monitoring these large spatial and bathymetric areas. We likely need an increased number of samples, primers, and or volume. I don't think volume was really mentioned throughout this conference, but in the ocean we are seeing that it is making a large impact the more volume you get, and it might be more important than replication. Sampling should also be weighted based on different water masses, while also being spatially stratified based on our GDMs. But also, I didn't really touch on it here, or I mentioned it briefly, that we still have a lot of unassigned ASVs. The ocean is vastly understudied. I think we know more about our solar system than we do the deep sea. So really, we need this more comprehensive reference database, and I talked to Genevieve about it. Maybe it might become one of my chapters. But we really need a better reference library, or even just a regional reference library, to make better detections because of the number of unassigned ASVs. And this is something I've talked a lot to with DFO, is to kind of make some standardization between research groups. It's something that's really necessary, but also between universities and government, because we're sampling the same areas. Let's make one massive data set that we can compare over time and between each other, rather than doing different things. So we're hoping to get a renewal on our grant. And then moving forward, I hope to standardize those methods between the two research groups, between mine and at least DFO in Newfoundland. All right, with that, thank you everybody who's made this work special. Monitoring at sea or collecting samples at sea is not a one-person job. So my supervisors, John Fisher and David Cote, but also here today, Genevieve Parent and Audrey Barrett, again with the bioinformatics. So thank you everybody.
Hi everyone, I'm Sam, and today I'm going to be talking about part of my ongoing PhD research, where I've been working to build a citizen science network in Nova Scotia. But to situate my work geographically, Nova Scotia is part of Mi'kmaq. This ancestral and unceded territory of the Mi'kmaq comprises present-day Atlantic Canada, with Nova Scotia making up this peninsula here. As you can see, it is almost an island, connected to the rest of the mainland through a very narrow land bridge. And so despite its small size, there's a lot of coastline in this province, over 13,000 kilometers. And as this province touches upon multiple oceanographic regions, it is home to diverse marine biodiversity. And so perhaps it is no surprise that the inhabitants of this land have always had a very strong connection to the ocean environment. First culturally, but then eventually economically as well. Perhaps it's no surprise to hear that this connection is threatened. We know as a result of anthropogenic climate change, we are seeing impacts in the ocean. There are too many to name, but a few relevant to the context of my work include an increase in sea levels, which can threaten both coastal habitat as well as coastal communities, the acidification of the water, as well as an increase in water temperatures, with this last one being of particular concern in the part of the world where I live and work, as the Northwest Atlantic has been observed warming at rates about 3 times faster than the global oceanic average. And as a result of increased water temperatures, there's this global trend of species appearing to move towards the poles, chasing water that lets them stay within their thermal preference. An example local to Nova Scotia is that of the silver hake. This is a small fish that is historically fished in and around the south shore of Nova Scotia. But over the years, fisheries landing records and trawl survey data suggest a decrease of biomass in this region. Meanwhile, nearby Newfoundland has been reporting increases in biomass of silver hake. And so this appears to be a local example of that northward movement. And it's one thing if we have decades worth of fisheries or trawl survey data to look upon and detect these trends. But if we want early detection of this sort of climate-related movement, we're going to have to be consistently looking for it, which perhaps no surprise is often very challenging, both due to the time and expense involved. However, citizen science offers a potential solution to mitigate some of the challenges associated with ongoing monitoring of marine animals. And so, the research objective of this chapter has been to build an eDNA-based citizen science network in Nova Scotia for ongoing monitoring of coastal marine animals. This network is called the Community-Oriented Coastal Observatory, or COCO, as I will be calling it going forward. And how COCO was conceptualized to work is that we begin with our citizen scientists. These are the people on the ground collecting the eDNA samples through filtration. After they collect those samples, they then pop the filters in the mail where I receive them at Dalhousie and do the lab and bioinformatic processing to determine what species contributed DNA to our sample. I then share those results directly back with those who are collecting the samples in the form of a species detection list, as well as posting it on the COCO website, so anyone who's interested can head online and see what species we've been detecting around the coast. This sampling then repeats, seasonally 3 times a year. And so once we had the workflow of the network figured out, it was time to actually turn it from an idea into a reality. And this development took 3 main steps, which I will briefly touch upon each in turn, beginning with our site selection. I ended up picking these 6 locations along the coast of Nova Scotia as my initial COCO launching points. There were a few reasons why I chose where I did, mostly logistic. First of all, I had 6 eDNA samplers at my disposal, so I was going to be limited on the number of sites I could begin with. Second of all, I targeted communities that had a public library. This was done for 2 reasons. Firstly, it was my chosen forum for the initial outreach events, and secondly, it was my first choice for a community home for that eDNA sampler itself. And lastly, by situating all of our sites along the same coast, we don't only want to compare species detection within community, but also between communities, and see if we can detect any of that northward movement over the time series of the cocoa sampling. And so once I had picked these sites, it was time to actually go there and see if the communities were interested in participating. And so this did take the form of a series of library outreach events, where I just went into the community and I gave a presentation very similar to the introduction that I went through with you guys. So I just explained the rationale of the network, what was going to be required of volunteers, and then we had a conversation. What brought them out, what changes were they seeing, and where in community would be a good location to actually do the eDNA sampling. While I was in each community, I did stop off and collect 3 eDNA samples, as I had never been to any of these sites before launching the network, and so it served as initial site validation, as well as starting to get some baseline species data. Of the volunteers who then said they were interested in joining, a few months later we went out to the coast together and did some hands-on eDNA training. So I demonstrated how to set up and use the machine. For our purposes, that is the Smith Root Citizen Science Sampler pictured at the top of the screen there, as well as how to assemble the filters. Once again, these are Smith Root, how to collect the samples, and then how to preserve them. You can see the photo at the bottom there is me demonstrating how this worked. And together we collected 3 eDNA samples per community. I do want to note, attendance to this training session was not a requirement to be a volunteer, as we just picked a day that worked for the majority of interested people, and we do provide other instructions to people who join the network and did not have an opportunity to participate in the training. And what was super exciting is while we were doing this, DFO was actually present at one of the locations doing some seine netting. And so we've got visual confirmation of the sort of species that we are expecting to detect through the cocoa sampling, notably this short big eye. This is a reef-associated fish that is typically found in like the mid-Atlantic coast of the US down into the Gulf. So it is not known to live in Nova Scotia, and yet DFO caught it. And these are the sorts of species that we anticipate detecting with more frequency as the waters continue to warm around Nova Scotia. And then March of this year was the first time that the volunteers went out and collected eDNA samples on their own. So I mailed the consumables directly to one of the community volunteers, while another one picked up the eDNA machine from the community home where we had left it after our training session in the fall. And I'm super pleased to say that all of my volunteers were successfully able to collect and return 3 eDNA samples, and these are just some photos showing them in action. And so the current status of cocoa is that we do have a few seasons of samples under our belt. However, I have only started to analyze the summer, while the fall and winter samples are currently waiting to go on the sequencer. Next month is going to be our second fully volunteer-run sampling event, and then the plan is for cocoa to continue at this frequency throughout the duration of my PhD. But ultimately, the goal is to have this outlive my studies and to continue well into the future. And so as I mentioned, I have preliminarily analyzed the samples collected during my outreach event last summer, and so I would just like to go through those and show some of the baseline data that we've started to collect. On the left side of the screen is a very brief summary of the methods that I used to determine the species detections on the right. To read this plot, the community that the sample was taken in is along the bottom, while the different fish species we detected are along the top. And if you follow the ribbons between those two sides, you can see what we were detecting where. Of those 26 unique detections, 5 were ubiquitous, and these are Nova Scotia classics, so it was absolutely no surprise to see them all along the coast. What did jump out to me was the presence of these 4 freshwater fish. Notably, 2 of my community locations were very close to freshwater outflow, and as a result of this, those sites have since moved to another location within the community. But my lab also does do a lot of freshwater eDNA work. And so this could represent lab or equipment contamination, especially as freshwater samples were included on the sequencer during this run. And so I'm hopeful that analysis of subsequent samples will help inform where these detections came from, and that by moving our site to a different location, we can try to not have this happen in the future. And meanwhile, while we've been doing the field work, I've also been testing and validating the methods. Namely, I'm super curious about what's happening to the DNA on that filter between the time the volunteers collect it and I receive it in the mail at Dalhousie. And so this validation is taking the form of what I'm calling my time to extraction experiment. About a month and a half ago, I went out and I filtered 72 eDNA samples all at the same time. I filtered these onto 4 different combinations of filter material and preservation method. So half of my samples were on the PISCeS material and half were on mixed cellulose ester. Of those 2 material types, half of the filters were preserved with silica beads, so desiccating beads, while the other half were preserved with Smith Root's proprietary self-preserving filter. The majority of the water that I was filtering, about 99.9%, was coastal ocean water. But I did spike in water from a tank of captive Atlantic whitefish to act as a positive control, so I could perform qPCR immediately after extraction, confirm extraction success, as I will not be sequencing these until the final extraction is done a few months from now. The goal is to assess the broader fish community composition, but as I have performed the analysis of the first 4 extraction points, I'll just quickly walk you through that qPCR data. So along the x-axis is the day that the extraction occurred after the sample was collected, while the y-axis is the concentration of whitefish DNA I was detecting. The concentration is a bit all over the place. I think, given the nature of eDNA, not a bit surprising. So rather than paying attention to the concentration of eDNA, we're just going to look at the detection patterns right now. And so my first 2 time points, which occurred day 3 and day 7 after collection, all of the combinations I was testing successfully returned DNA of that whitefish. By 2 weeks after collection, I no longer had whitefish DNA on my PISCeS filters, and by 28 days after, I only was having detection on the cellulose filters, which had been preserved with the silica beads. And admittedly, this is early, and I don't know how well the qPCR data is going to track the broader fish community composition, but a few early takeaways for me are that cellulose does appear to be better suited for the marine environment. Secondly, DNA loss between collection and extraction is a concern, particularly when it's DNA of species that we've detected in a low abundance. And as a goal of COCO is that early detection of climate-related species movement, this is important for us to know. And so as a result, I am now asking my volunteers to return their filters as soon as possible, whereas before I gave them a little bit more leeway. And so with that, the significance of my work comes down to the fact that we know the climate is changing and that species are going to be changing in response. And long-term monitoring is very valuable in the long term, but it can also provide very important information in the short term as well. And quite frankly, citizen scientists are awesome. There's so much knowledge and interest within community. They've been invaluable in terms of site selection. And by partnering with the people that already live in these areas we're interested in studying, we can sample a larger geographic area and at a more consistent frequency than otherwise possible. But of course, when trying to establish something like this, the methods need to be validated for the study system that we're working in. And so with that, if you want to know more, shameless plug, but a paper about the development just came out on Monday. So it's very hard to condense 2.5 years of development into 12 minutes. And there's so many people that made this network possible, this project possible, but I just want to give a shout out to the volunteers. This sampling does not happen without their engagement, their time, and their effort. So thank you so much for listening.
Thank you very much. We do have time for one question.
Thank you for your presentation. I have a question regarding the self-preservation Smith Root. This is a really good method to collect the samples, but I haven't been convinced that it's the best way to preserve the samples. And you showed that after you test the samples after a couple of days or weeks, the detection rate for the silica beads is higher. Does this mean that the samples is degraded on the self-preserve?
That is what the early data is suggesting, but again, this is DNA that was already existing in a low concentration, so I don't know how well this would be extrapolated out to a species that were detecting a lot of their DNA. I have heard other researchers have good luck with the self-preserving filters, but it does appear for my purposes and detection of those rare molecules, it might not be the best if you collect it and then leave it on that self-preserving filter for 2 weeks or a month.
Thank you.
Thank you very much.
Hello. It finally gets to be my turn to talk when I'm at the front of the stage. I'm excited today to share with you all a project that we were able to get published in the journal Environmental DNA just at the tail end of last year, entitled The Congruence of Fish Community Diversity and Composition Estimates Using Water and Sediment-Based Environmental DNA in a Recently Restored Creek. So to begin, I'm sure all of us in this room are very familiar with the fact that comparisons of conventional sampling methods and eDNA-based methods are incredibly common in the literature. In small streams, which are of interest for this study, are typically comparing electrofishing with aquatic eDNA. And there's minimal literature comparing sediment-based eDNA methods with conventional methods. And a review paper was published early this year exploring the just sheer number of comparison studies with eDNA and conventional methods. But what I want to draw your attention to is that this large green area at the top represents the comparisons of electrofishing with aquatic eDNA, and that teeny tiny little sliver that you see with the red arrow is pointing to those same kinds of comparisons with sediment. And you can imagine even further, fewer studies are comparing multiple of these methods, whether multiple conventional or multiple eDNA-based methods, simultaneously in the same study. Now, why this far into the development of eDNA should we care about comparisons? And it's in big part because regulatory obligations rely largely and almost exclusively on conventional assessments. But there is interest in the use of monitoring approaches that less directly impact organisms and the environments that are being monitored. Additionally, as we've had a lot of conversations about today, contextualizing eDNA data and eDNA findings with trusted methods that people already believe in can make it easier to facilitate the development of further trust in molecular methods. And we've had a wonderful panel discussion earlier where I think a key theme was that sometimes the biggest barrier can be the trust in uptake of the methods, not the actual feasibility or correctness of the data and interpretations that we're getting. And so to scale back into our specific study, when we think of eDNA that is present in streams, we obviously think about aquatic eDNA. This is DNA that is recently released into the water by organisms, and it can be relatively stochastic. So you can have DNA become present after a very short period of time, maybe a fish or another organism is transiently passing through and they release a little bit of eDNA once, or maybe they're releasing it constantly into the environment because they live there. Additionally, we have benthic surface sediments, and these can represent a longer term average of accumulation of DNA that is settling out of the water column. And it tends to be a little bit less stochastic because you have to have a prolonged presence to have enough DNA that survives the process of degradation and sedimentation to begin accumulating in that sedimentary layer. And additionally, we're interested in a sort of in-between sampling medium that we refer to as suspended solids. And so these are particles in the water column that are still relatively recent. Maybe they have yet to sediment into the water column, or maybe they never will. And these reflect areas where DNA may be shed into the water, and they're able to bind and start getting preserved accumulate over time. So they sort of represent this in-between of freshly shed aquatic DNA and longer preserved sedimentary eDNA. And so we had an opportunity to ask a question. Is there a single eDNA medium that most similarly estimates freshwater fish community diversity alongside electrofishing, so that we could start to develop some more of the trust that I was talking about in some of these sedimentary eDNA methods? And so we had an opportunity to explore some neat questions like this at Martin Creek, which is a little creek just north of Guelph. It has an interesting history where between 1832 and 2010, it had been dammed, and it was functioning as a warm, still water pond. And in 2010, a group at the Ignatius Jesuit Center and other stewards of the land came together to take the dam away and restore the creek into a flowing cold water creek. And so you can imagine that a very substantial transformation of the expectant biodiversity in that area will have transformed from 2010 onward. And there was interest in seeing if we could use eDNA methods alongside conventional methods to see how things have been recruiting into the environment since the dam was removed. Additionally, because we have this known big impactful event in the environment from 2010, we know that that very topmost layer of sediment in the water is going to be reflective of whatever is colonizing or recolonizing that aquatic habitat since the change in the habitat type. We collected at 4 sites. Only 3 of them are visible with the dots because of the angle of this photo I was able to find. But we have 2 on one side of the river, and the river, or the creek, sorry, is intersected by a highway. And so we collected 2 on each side of the highway, and we collected samples once a month between September and December in 2021. And all of the field work and much of the lab work was conducted by an excellent master's student that we had in the lab, Erica Myler. And at each of these sites and sampling times, we collected filtered water from the flowing water in the stream using an osmosis sampler. We collected 2 liters there. We also deployed these custom DIY sediment traps that Erica and Gerald Tetro worked together to develop, where essentially water will flow through them along the bottom of the river, and all the particles in the water will, not all, but particles in the water will flow through it, and they won't be able to leave it, and they will get trapped inside of this tube, and then we can go and recollect that PVC pipe in the water and pull the sediments that have accumulated inside of it. We've also collected bulk sediment from the very top layer of the sediment in the creek. And then, of course, to have a conventional method to compare against, we conducted backpack electrofishing. Once we took those samples from the field into the lab, we extracted them based off of their sample types. So we used 1/4 of a piece of filter paper using the blood and tissue kits for our water samples. And then we used the Power Soil Pro kit for our sediments. We used the 12S MyFishU marker. We sequenced using an Illumina MySeq platform. And we also included a mock community positive control that included 23 expectant or likely or confirmed species of fish, including DNA extracts that were derived from fin clips from individuals that were captured during electrofishing. And then we used a custom curated reference sequence library to make all of our identifications against. So we made a list of Ontario native and potentially invasive species. And we used a mixture of public data as well as some of those sequenced local haplotypes that I just mentioned. And in total, we were able to create a reference library that was representative of a little bit over 78% of all this freshwater species that we know to occur in Ontario. And we don't expect all of those to occur in Martin Creek, especially because it's so recently transformed, but this allowed us to cast a wider net, so to speak, particularly when it comes to the potential for false positives. And then last, we used the bioinformatic pipeline BARC to process all of our data, and we used a threshold-based read cutoff of about 0.05% of the mean read count across all of our field samples, which ended up being around 17 reads, because this gave a relatively informed cutoff to use based off of the read depth we were able to get across our study. And so, the first measure that we looked at for this comparison was alpha diversity through species richness. And what we found was that the trapped suspended solids estimated the most similar amount of richness to electrofishing. And I'm not expecting you to read all of these species, but these were all the species that we detected over the course of the study, separated by method. And what I want to draw your attention to is the fact that water estimated the highest level of richness, and all of the trapped suspended solids detections are a complete subset of the species that were detected by water. Next, we looked at measures of community diversity through analyzing Jacquard's dissimilarity. And what we found was that all of the eDNA methods generated estimates of fish community diversity that were more similar to each other than any of them were to electrofishing. Additionally, we found that site had a significant but relatively much weaker contribution to similarity with respect to the fish community estimates that we saw. So it had some effect, but it was much less impactful than the sampling methods used. And last, the sampling month had no significant effect on the fish communities that we were observing. To explore that a little bit further, we also conducted an indicator species analysis. And for those who are not familiar, an indicator species analysis uses a measure of specificity, so how uniquely a species is associated with one particular method, and a measure of sensitivity, so how frequently a species is found with that particular method. So as an example, a species that's only ever found with electrofishing, but is only ever found at one site, would have a really high level of specificity for that method, but a really low sensitivity. And this analysis is trying to find species that have a high specificity with a method, as well as a high sensitivity, to make that strong correlation that that method is most able to capture, or most sensitive to detections of those species. And what we found was that, not entirely unexpectedly, there is a mixture of species that are being preferentially detected by electrofishing, some that are being preferentially detected just by aquatic eDNA, and then a couple of species that are being found by eDNA, but it didn't matter which sampling method that you used to collect them, and then a large overlap there in sort of the gray in the bottom, where it didn't matter what method you used, they weren't significantly associated with detection by any particular method. And something that I think is particularly interesting, and that Charlene sort of highlighted, that you shouldn't ever assume that what you find is true from the jump. But we found that all of the taxa that were associated with electrofishing were not able to be identified in our mock positive control, where we know that those species' DNA had been included in the sample. And there were sort of two ways that we couldn't get a detection. Either the DNA was not amplified and confirmed, or DNA maybe was detected, but it could not be resolved uniquely to species. And when we looked at our data more specifically, we were able to identify that we weren't able to resolve some of the most common haplotypes for species belonging to sculpins, so the genus Cottus, as well as several species that belong to the family Luciscidae. And this was not surprising, because these are groups of taxa that are well documented in the literature as having high rates of hybridization, morphological similarity. And so there's a wide range of reasons why we might not be able to discriminate these species from each other. And I just want to highlight as well that two of the most unable to be resolved taxa were also some of those taxa associated with just electrofishing. And so the takeaways from this study was that no single eDNA method that we used was most similar to electrofishing. The eDNA methods were all most similar to each other. Sampling site and time had little to no impact, respectively, on the differences that we observed relative to the large impact of sampling method. And the dissimilarity that we observed appeared to be more driven by technical limitations related to taxonomic resolution and primer-related species detection biases. And it's not likely that the inability to capture these species was because we weren't recovering their DNA in the samples. And with that, if we have time for questions, I'm happy to take some. And if you would like, because we were able to publish this study relatively recently, if you scan this QR code, it will take you to an open access article that you're able to read on your own time. Thank you so much.
Yes, we have time for one question.
Did you separate the extracellular and intracellular DNA in your samples?
No, that is a good question. We did not, which can be particularly important for sediments. It was one of our first forays into working with sediments in the lab, and we had a lot of difficulties getting anything out of the sediments. So we kind of got the best that we could out of them.
I have exact same experience.
Yeah, it seems to be quite common with sedimentary DNA. I will plug Shu and Nicola from Lab Services. They were the ones who were able to get the DNA out of our sediments, and we would not have been able to do it without them. So.
Thank you. One more time for President.
Okay, so hi everyone, so my name is Francis LeBlanc. I'm a biologist and lead an eDNA testing lab that we have within Fisheries and Oceans Canada to support the management of aquatic invasive species. So I'm going to take the next 10 minutes to kind of present the work we do, how we operate, as well as some of the implications of operating under an ISO 17025 accreditation. Okay. So I likely didn't need to add the first bullet point for this audience and at this time in the conference, but for those who don't know, Fisheries and Oceans Canada, so we're a Government of Canada department, so a federal institution responsible for the stewardship of our fisheries and marine resources, along with other levels of government. So like many here, we have early on started using research, doing research and development on eDNA for macroorganisms in the early 2010s to try to see how it compared to other more conventional methods and how it could help to manage some of the issues we deal with. And more recently, there's been willingness to start adopting it to support decision-making process, compliance. And as we've heard earlier by Chris, aquatic invasive species are one area where eDNA is well suited to help on that behalf. So what you're seeing here is kind of the 4 pillars that DFOS follows in terms of AIS management, all the way from prevention to control management to try to manage these invaders. So in terms of prevention, you can take the example of eDNA being used for fine-scale resolution of the distribution of these species to help, for example, with the risk assessment in terms of introduction and transfer decisions to support aquaculture activities, either marine or freshwater. And then on our early detection, I think it speaks for itself, the fact that it's quite sensitive compared to some other methods in terms of enabling us to detect them early so that we can take controls and management measures. And then on the very last of those pillars, eDNA could be used after eradication or control measures have been put in place to try to determine how effective they've been. And that's where we kind of heard eRNA being mentioned at least once this morning in terms of potentially being used to determine after eradication if there are still live organisms found in the environment. So as you kind of all know, invasive species can have major ecological and economical impacts. And I've just listed here some of the AIS that are in part true of the AIS regulations which came into force in 2015. So these are some of the species where control measures are currently in place in areas that the species are non-native. So you have, for example, some of the tunicate species on the very top, which are quite problematic for shellfish aquaculture in marine environments. Green crab, which again can have quite an impact on eelgrass in estuaries, which is a critical habitat for a lot of native species. And then you have some various fishes. And there are quite a bit more other AIS listed in Part 2 of the regulations which have actual their possession is prohibited in either all of Canada or in certain provinces. Okay, so with all of that said, because of the recognition that eDNA could be used to support AIS, there was willingness within the department to have some capacity to do some testing in-house. So because of that, in 2023, there was some funded directing to establish a national lab within the department to support internal testing demand. So because of that, our lab in the Guelph region, so DFO has 7 Guelph region nationally. So we are located in Moncton, which is part of the Guelph region. And one of the main reasons for selecting our lab as the lab for the AIS support was that we were already doing disease testing as part of the National Aquatic Animal Health Program, which is co-delivered by CFIA and DFO, and that was an accredited lab. So we were able to leverage what was already built as a quality management system for the NAP program and apply it to eDNA work for aquatic invasive species. And I'd also like to highlight, while we are officially recognized as within the department as being the testing lab for AIS, there are quite a few other genomic labs within the department that play an important role in AIS eDNA work, either on the research side or on the testing side. And there is still a lot of collaborative work with provincial labs and private labs, because the demand year over year continues to exceed what we have in terms of capacity. And another argument, I guess, towards having more labs to be able to accommodate that demand we have nationally. Okay, so for those that might not be familiar with what the ISO 17025 standard are, so they're not eDNA specific, they're just general standard for laboratories doing testing or calibration to demonstrate technical competency. So non-related to eDNA at all, but they apply to be able to demonstrate competency, and it generally helps support validity of the results. That is sometimes required for regulatory purposes. So what we currently have on our ISO scope of accreditation is only the targeted qPCR. So most of the work we do is targeted qPCR, which is what is on our scope of accreditation, but we also do a bit of metabarcoding, but that is not on our scope at the moment. Okay, so what are the implications of operating under an ISO accreditation? So it mainly has to do with having a robust, what we call quality management system, which lays out both management requirements and more of the technical requirements. So on the management side, there's everything from good document controls, having the right SOPs, having what we call corrective and preventive action. So always looking for areas for improvements, having non-conformance when errors happen. There's a lot of documentation that needs to take place to ensure that you're in line with what the requirements of the standards are. And then there's the internal audits to demonstrate that you're continually improving. And on the technical requirement, we have staff competency and training, and this is where the proficiency panel come in. So at the moment, we mainly rely on interlab validation as well as in-house prepared proficiency panel to demonstrate that staff is competent. But I'm glad to hear that there is work towards having potentially external panels available for labs that wish to participate. So that's going to be great. Method validation, we mostly follow the CSA standard for targeted qPCR work in terms of in terms of ensuring our tests are fit for purpose for the work we're using them for. And then there's measurement traceability, making sure that the equipment are performing as expected, and then all of the SOPs regarding the protocols that we follow. So this is just an AI-generated workflow that kind of applies to all labs doing eDNA targeted testing in terms of all the way from this field sampling to the reporting results. And everything in between, right? In terms of having a good documentation system for sample traceability from the time you do receive them all the way to the results reporting. I have highlighted the sample collection category in green just because that's outside of our accreditation. So our accreditation covers from sample receipt to the reporting of results. And I guess with ISO, it emphasizes the importance of having the right QA/QC checks to make sure that you know where the uncertainty is in terms of the false positive, false negative risk all through the lab. Just wanted to go a bit into more details about the specific of what is commonly done within our lab. So for the sampling portion, most of the times, our field program collaborators will go with triplicate samples, 1.5 to 2 liters, generally collected with in situ sampling approaches, Haltech or Smith-Root. We mostly use glass fiber filters, 1.2 or 1.5, but again, this is sometimes adapted as well to the purpose of the work that's being done, like we heard this morning, right? If it's more of a presence type of of sampling approach that's being done. And then DNA extraction we're mostly using, as most of you are, some commercial kits such as the Kaigen DNeasy. We actually use a T4 bacteriophage as our IPC that we spike in at the DNA extraction step. So it is a process control for both the extraction and inhibition. And if there is an issue with our IPC, then we can go and determine, okay, is it qPCR inhibition or problems at the extraction step. Most of our testing is done in duplex, including that T4 IPC, and then we mostly do triplets. I won't go into details too much because Genevieve is going to talk about this in the next presentation, but this is quickly how we report qualitatively results. So we have the not detected, no amplification. Inconclusive is when there's no repeatability. Detected when repeatability is there. And then unfit is what we use when the IPC didn't perform as expected and there's a higher chance of a false negative. So we'll relate to our colleagues that it was not detected, but because of the higher chance of a false negative, we've started using that unfit category for those. We currently have 35 targeted assays operational, 22 of those are for AIS, and we are currently working on designing, making operational another 8. And I will say that from these lists of 35s, some were in-house validated, but others were taken from the literature, and we just tested them to make sure that they work for where we wanted to apply them. Just a quick example of some of the eDNA work we support. So for example, we'll monitor for tunicate species to complement what is done with our DFO biofouling monitoring program to be able to assess where these species are for INT, like I said, decisions. And then as Genevieve said, we've been using them to see if mouse balls might be infested with ZQM to be able to quickly take actions because ZQM are prohibited in most provinces. So just want to finish off quickly by mentioning a few of the research activities that we are working on. So we are working on validating, making operational new assays, also on some targeted metabar coding to try to see if that could be at some point operationalized. Due to some of the limitations of qPCR, where we generally are limited on the amount of qPCRs we can run per sample, and we are asked to do more and more qPCRs on one sample, so there is somewhat of a limit there unless you want to start using those high throughput platforms. We already heard from Kaylee of some of the GAT eDNA, which is led by Anaïs Lacourciade, and then there was a booth outside regarding the portable lab fast eDNA that was led by Megan McBride with DFO in Maritimes. So I think that's it. Just want to quickly acknowledge a few of my colleagues. So Michaela was here at the last PISCeS presenting on striped bass overwintering work, and some here might recognize Nellie, which was quite active in the field, but is now retired. So she's still on emeritus status for those who might want to continue collaborating. So that's it for me. Thank you.
So hi everyone. Today I'm presenting what is an eDNA detection, which was a discussion happening between all this great group of authors. And you also see that in the title it's toward a definition. And so I'll explain to you what we ran into. And I'm not going to highlight that in multiple publications that we can read. There are a lot of people highlighting that there is no consistent definition for all of us. And so this is kind of an issue. And we ran in it with us, with a really big problem, as one of our colleagues, Megan McBride, in one of the region, asked us, like, what is a positive detection, guys? And across all of the DFO labs from Eastern and Mid-Canada, we all have different definitions. And we all have different decision trees. So earlier, Chris showed a decision tree, and we use all of those to make our decision transparent on what is or not an eDNA detection. But if you're working for the same department, maybe you should have a similar definition, for instance. And we were maybe the cause of that. And so, as some others have reported in the past, like we've published a document which is available freely online, where we said what are the minimum criteria you should use, or what are the items that you should report in an eDNA study or an eDNA detection report, so that we can use that and can be a reliable source for management. Because we were running in that problem with DFO management, so not DFO science, but the management side would receive, for instance, eDNA detection report and asked to take decision for mining or different under different contexts. And they were coming to us with those reports saying, what can we do with that? And most of the case, we could not understand methods or even how detections were done, or decide that it was a detection or not. And so that document, when we published it, we aimed at saying, be transparent and publish the way you are doing the things and taking decision if it's a decision or not, a detection or not. But we also left a lot of flexibility within that document, because research needs flexibility. And in 2021, we thought that that was necessary. But at the same time, we ran with that idea in our lab and then made our own decision tree, each of us. So after that initial discussion, we went on and said, okay, since then there was a lot of reference documents that have been published. Let's look at them and let's see if there are some definition within these documents that could be used by DFO and for us to have a more consistent definition. And so we use all of those documents and had a look at them. None of them had a one single sentence definition, but they all had points which we could extract and see similarities and difference. And those are the similarities, where all of the definition required an LOD and an LQ to characterize the assay performance, all required qPCR replicates to be ran on samples, all necessitated positive and negative control and an inhibition test, and all had positive, negative, and some intermediate classification to communicate the uncertainty behind some of the detection to their clients. And there were also major difference between documents. The flexibility, as I talked to you about, so we had the DFO document, which was more of a guideline at first, and then some other documents came later, such as the CSA we've been presenting since this morning, which was really a document presenting standards. And we also, for replication, there was a lot of different ways of replicating detection. And so, for instance, it could be advised for a greater number of qPCR replicates, but also a greater number of samples for you to be able to conclude to a positive detection. And some documents were using also LOD, limit of detection, or LOQ, limit of quantification, to consider if it was a detection or not. And also some documents required sequencing to consider if it was a detection again or not. So, okay, there are all these items, again, a discussion within our DFO family. Some position, like we were keeping some of us our position, which makes sense. And then we asked some more people to come in our discussion to be inspired. And so we contacted Adam, Karen, Guillaume, and Robert to come in and say, okay, help us to discuss that. And we also did something, because the discussion between us were going everywhere. And so we decided to narrow down the context of the definition, so that we all had in mind the same kind of studies that should be conducted. We were aiming to detect rare species, so DNA not abundant in the environment, such as invasive species in the early phase or species at risk, where we would like to reduce a maximum the type 2 error with false negative, because false negative rates, if they're high, then you're missing maybe the important moment to save the species or to remove it from the environment, given which status the species has. And also, we decided to focus our definition only on the lab proportion, so for qPCR replicates and also for samples that let's say, a lab like Francis is receiving. And so this is the part that we can control as a service provider, and so we decided to conclude on that. Also because if you're considering the field to say what is a positive detection, it's a lot of fun to discuss. And we also decided to focus on labs that had the experience to work with, or technicians that are highly skilled to work with eDNA. And I wrote monospecific qPCR detection, but probe-based also, so that there are standards available. We would not argue again on how the assay should be conducted, as these were available as standards. So it would kind of focus our discussion. So we had some discussion over around a year, not every day, you know, but like sometimes like in that year, thinking more and then coming back and bringing more arguments. And then we had a definition with agreements and divergences in 5 points, which I'm going to talk to you about. So for definition and agreements, we also thought that we needed positive and negative control that passed as any definition. We also considered that minimal inhibition needed to be detected to conclude to negative detection. For positive eDNA detection in a qPCR replicate, we needed a sigmoid amplification, not necessarily with a plateau, but that happens within 50 cycles. And that 50 cycles was kind of not discussed really, because all of our labs were doing 50 cycles. Since we're working with environmental matrices, we think that we need a higher number of cycles to avoid limitations in detection and favor amplification, given the matrix itself can limit the amplification. We also considered that we didn't need LOD or LOQ thresholds. And this was because we considered that those two measures are to assess an assay performance and not, they're not necessary to conclude to an eDNA detection. And we also considered that we didn't need Sanger sequencing if we were using a probe-based method. Because for all of us, like all of our tests that we ran, we never had like any non-specific amplification using a probe-based approach with a well-validated assay. And it's also because for Sanger sequencing, you often need a concentration higher than the LOD also, and most of our detection are lower than LOD in early invasion or with rare species, and so this is why. And where we agreed to disagree was for those 2 points. There was a lot of need still for flexibility in qPCR readability to conclude to a positive sample. And so there was a lot of variance within us, like on the number of qPCR replicates that had to be run for a sample. And this was most likely sometimes with program associated, such as PISCeS, which receives a lot of samples and has to limit the number of replicates if he wants to run to all of these samples. Or other labs where it's more cost-associated projects where sometimes there's very little cost. But a minimum of 3 replicates was again acknowledged as the minimum to do. And for the number of positive replicates, there was also no agreement. Some of us thought that a single one should be possible, the single qPCR replicate that is positive could be a potential positive sample in a context where there is no contamination within the lab, and you're convinced, because you've run this assay for a long time, that your limit of blank is still 0. But so for other labs, they were arguing that the contamination is random and can happen randomly, and then that this replication should be needed at the qPCR level to call it a positive detection. There was also some suggestion that it's also linked to project objective. In that case, I would be careful just because I think we need to think about this objectively within the lab, saying what are our capacity for detection. And we need to have lab arguments for us to conclude about the limits of or like where we can say it's a detection or not. And for classification of detection, again, we did not agree. So we kept those intermediate classification because some of the authors like them. We added a category even, which is suspected. And some of us didn't like those because our clients like better if it was detected or non-detected. So what is our one sentence definition? It's that one, where the first part is there is no flexibility, where to call a positive qPCR, you need a sigmoid amplification observed within 50 cycles. But the second part of the definition needs some flexibility. And this is where, again, replication and classification framework or decision flow should be reported so that it's clear for everyone. What was also interesting during this discussion is all of what we raised as concern where there is no clear definition. And for instance, what is inhibition? Like a lot of us have different definition for what is inhibition in qPCR assay, although there are standards, but there still needs to be precision. What is a positive detection using metabarcoding? This is a really interesting discussion to have with, that I had with some of you. What is a positive detection considering the sampling design? This is where some of our colleagues were like, well, you should consider modeling. If not, it's not really interesting. So, interesting position. And what is an optimal field control? So, again, there are a lot of questions raised. At some point that, although there are standards moving forward and it's interesting, sometimes in those standards we need to move fast. Within 6 months, we need to have an answer. Maybe those questions and background could be discussed with a group of scientists and then their opinion could be published. And we were thinking that maybe a virtual collection of those questions that would be tackled between a bunch of inspired scientists could be interesting to publish. So we would have like a kind of a state of the field in 2026 and 2027, and then those definitions could go further or be pushed forward in the future. So if you're interested, just let me know. We can move on with maybe a virtual collection or a special issue on that. So thank you for listening.
As we head into the last session for the Act 3 of PISCeS, I'm pleased to introduce my friend and colleague, Sujeevan Ratnasingham. He is the architect of the Barcode of Life data system, former Eddie Nielsen Prize recipient, and we could go on about that. No need to embarrass him. But this is going to be an interesting talk about the potential role for using large language models in AI in supporting our research. So I think you'll find it interesting.
Thanks so much, Bob.
If you want, we've got a laser pointer, and then it should.
Should advance?
Yeah.
Perfect.
Okay.
Thanks. Yeah, I apologize for being the last class of the day. So I'll try and keep it interesting and quick and light. But I have to thank Bob for the last minute invitation. So pleased to be here. I was here for one of the past PISCeS conferences. Fantastic event. So I'm pleased to return. I'm talking about, as Bob said, using large language models, and I'm sure everyone's familiar with them. I started working with large language models and generative models about 2 years ago, and the real driver for me was this field, where the number of papers in this field was just ballooning, both in biodiversity, eDNA, metabarcoding. As you can see from the trend, it's more papers than one can read, but interestingly, this sort of volume is also creating a lot of pressure on the peer review system. And so having the ability to employ discernment when doing meta-analysis and extracting knowledge from papers is really important. And I wish I could say generative models to the rescue. It's a great solution, really exciting, interesting time to be in data science. And it addresses the two big problems that we have, which is the flood of data, but also the knowledge access gap, which is that even today, even though databases are prominently used, there's a ton of knowledge that gets buried in literature. Fortunately, more of it's becoming open access, and large language models have been incredibly popular and serving a broad range of needs. These are a collection of what's called the frontier labs. These are the closed solutions that are out there, mostly commercial. OpenAI leading the charge, Anthropic now taking the lead. I list two numbers there that are really, really important to keep an eye on. One is the number of parameters, and these are estimates because the frontier labs don't disclose these values. The number of parameters give you a sense of the knowledge that that model has encoded in it. And the context is the amount of input that it can take when generating a response. And those two numbers keep growing, which is the indicator of the power. It's not whether it's called fable or mythos, but the size of the model and the architecture of the model, which none of these guys disclose. And all of these are powerful solutions. There are all sorts of pitfalls in their use in science, and well documented, and more meta-analyses, and editors' cries for help and looking for solutions in what's coming. But there are three risks that I want to highlight that need to be addressed when using large language models for the purposes we'd all like to. One is the hallucination risk, especially hallucination of citations, lagging knowledge. It takes a lot of money and a lot of time to train models. And so the models that we use are working with data from a year ago, or 6 months ago, or longer. And we have a sense when we use these models that they're recent because they speak with such confidence. So that's a challenge. But then there's also a risk of the dependence on the frontier models. These frontier models are heavily subsidized, and they're designed for a one-size-fits-all. And more, they're shifting to be more appeasing and telling you that your ideas are good, even though they may not be, or not supported by evidence. So these are issues that need to be dealt with. And one way to address that is with open weight models. And these are less well known. Hopefully folks have had a chance to look at open weight models. The one that everyone's familiar with is DeepSeek. And so these open weight models are models that have the model layers and the structure and the matrix weights fully exposed and modifiable, which means their training can be extended and weaknesses can be addressed. They don't have the same kind of investment that the frontier labs do, but they provide a really nice opportunity. And what's more, some recent advancements, recent in AI scale could be 3 months ago, 6 months ago, is what's called thinking models, and that thinking should be in quotes. Really, it's a technique called chain of thought, which allows a model to interrogate a problem through multiple iterative cycles. For example, you put in a question, 6 diners, 2 tacos, tortilla packs a day, how many packs? On a single one-shot response, there's a much greater chance that it's going to provide the incorrect answer. But chain of thought mitigates that by interrogating the knowledge base, if you will, which is the model in itself. So it's a multi-step reasoning, quote unquote again, error detection, and much better planning and use. One of my favorite models, which will run on my laptop, is the Qwen 3.6 27 billion parameter model. And with thinking models, there's a subtle shift. It's less about how large the model is. It's more about how well it can reason. So this 27 billion parameter model, which is now very popular, can reason without knowledge, because reasoning is a way to deal with new information that comes in. And so that provided a really useful opportunity. Many researchers, AI researchers, have jumped on this. And what we find is that reasoning pays off in certain contexts, especially mathematical and symbolic reasoning and logic. And that naturally fits into scientific work. I wouldn't say scientific work is common sense Knowledge, yes, in terms of terminology and definitions of terms that you find in literature, but symbolic logic is how you figure out whether a species is a species in a given area, whether it belongs there, or it's just an arbitrary relationship. So by putting together the various elements in the corpus. So the third problem of hallucination can be addressed using a couple of different methodologies. So with these open models, there's less reliance on the frontier models, less of a reliance on the one-size-fits-all. You can retrain a model. If you have massive infrastructure and time, you can fine-tune a model, which is much faster and far more effective to address the areas where it's going wrong. And you can use a method called retrieval augmented generation. And retrieval augmented generation is where you can take context, especially if it has a large context window, and put the background information in with the question that you have. And that becomes a complement to the existing model, which is very useful. And so I started down this journey to see what could be done. You know, how far could this be pushed? Specifically for this community, building an intelligent literature system. I thought funding would be barcode oracle. You know, everyone wants an oracle to go in and ask a question. There were 3 requirements that I started with, which is a cited corpus. The prose has to become queryable, it has to be structured, and you have to have traceability. And so an architecture started to form through multiple attempts. The base of that architecture is really important because today you could just take a whole bunch of PDFs and you can index them and you can pose a question. But that's not the same as having a model understand those documents. That's a search and retrieval. Although the frontier models employ RAG, where they can do web searches and pull things in. But what I was looking for is something that would be focused on the literature, exclusive of all the other information that was out there. And so to build a corpus, you need an ontology or a collection of ontologies, taxonomy, geography, traits, publications. And something interesting had to be done with the publications because there's so much text. They need to be summarized. So that was the first step, is deriving information from the publications instead of just indexing them. And then adding a fine-tuned model, a small model, because we want this not to be encumbered by all the training that comes from the internet. And so the fine-tuning only serves one purpose, to add definitions for terms. You just need to know what a species means. You just need to know what an aliquot means. You need to know what water density means. And so the ontologies extracted from the various databases get loaded into fine-tuning. And then you construct a knowledge graph. And a knowledge graph is a really old technology, which is where you define knowledge as a combination of triples, subject, object, predicate. You can define anything that we know in a set of those 3. It's a really verbose way to describe any knowledge, but it's something that's computer friendly. And then you need an agent. You need an agent that can ingest all that information against an inquiry and provide a useful outcome. So the process of generating the corpus itself was a pain, starting with open access literature from PubMed, using OpenAlex and the various PubMed resources to pull that down and deduplicate it. And you know, no surprise, when you search for DNA barcoding, metabarcoding, and eDNA, you get a bunch of false positives. So the first step is using a small model to classify based on the abstract whether you need it or not. And so that creates a filter. PubMed is beautiful in that it has an XML version of each document, which is super computer friendly. And so that gets pulled down, everything gets converted to Markdown. So the Markdown conversion will go from PDFs to Markdown or XML to Markdown, which is the language of large language models. And then we start the enrichment process, which is where we summarize the document and we extract the triples. And you can get hundreds of triples from a manuscript. And that gets imported into a SQLite database. and embeddings are generated. So embeddings really are taking sentences, converting them to vectors relative to an established meaning for words in a specific order, which is what large language models do. Once that's built, and this is where we are right now, took a long time to ingest this. So currently I've got 7,770 papers indexed, stopped at a nice round number, but that turns into 2 million extracted triples, 14 different extraction methods to get to that, and 156 different entity types. These were entities, definitions, if you will, that were used to generate fine-tuning. You get a bunch of authors. So once that's in place, you can run a query. And that query itself is a lengthy process. First, you parse the question. You've got to understand the question using a small 8 billion parameter model. And that initial parsing gets converted into vectors, which you can search for related papers. So what it does is searches for a small number of papers that are related to that question. Those papers are extracted and they're ranked via the RFF fuse, which is just a query ranking. And so you get the top 5 papers. And you go from the top 5 papers to the knowledge graph. Now the knowledge graph is this connected network that tells you what's related to what. You can use the knowledge graph to get other papers. So this becomes a choose your own adventure series. You get other papers and you start to get a mini corpus of knowledge that's related to your question. And that gets loaded into the model. And you use those seed papers to get your corpus. And then once you've got your seed papers, you expand that, as I mentioned. And there is a threshold because the way it searches across papers uses a very simple 0 to 1 metric for is this appropriate or not. Cutoff is used, and that data is fed into final evidence. And then the question is asked once again, this is where the agent comes in, and a response is assembled throughout that process. And what we want in the response is a very simple question like this. We want to have a series of statements that are logical in order, that are sourced in the way that scientists would be looking to source them, which is any statement that requires support will have a publication that explicitly supports it. In fact, within the system, it maps the support to a paragraph within each one of the publications used to support it. And I just recently added another mode because I noticed that sometimes there would be gaps. There's a deep mode where once it generates that response, it feeds that response back into the front end along with the question and says, what's missing? And you can't use the papers you used to come up with this answer so that you can find contrary information to that. And it works remarkably well. I've not made it public as yet. Happy to share it with anyone who wants to try it out. I love feedback. The problem is, whenever you're using AI, it's expensive. The token cost is quite high. So I won't tell you how much I've spent so much in token cost thus far, but it's substantial. And so I've got a lot more papers to get through because, as you saw from that growth graph, this should be up close to 30, 40, 50,000 relevant papers that need to be ingested. But as it stands, the interface organizes manuscripts by authors, so you can go in and pull up the paper, or by institutions. And for every paper, you can inspect how the system, it has to be transparent. So for every paper, there's a summary, and you can see over there for that particular paper, I picked a random one. It's summarized in a very structured way. The study organisms, the geographical scope, the scope of work, the findings, the challenges, the innovations. And so that summary is much lighter than the original document, and that helps speed the inquiry before you go do a deep dive. Over on the right, you've got the full text, but the very right is a table of all the associations. I can't zoom in on this, but I'll zoom in on one thing that you can do on this interface, which is to actually look at the graph. And so in this case, I don't know if it's readable on there, you can see that we've got a taxon, and we've got very standard predicates in terms of that taxon's relationship with other terms. And this is just one paper. So when combined across multiple papers, this becomes a scaffold for evidence that prevents hallucination, which is really important. It is the challenge that the LLM has to face when going through this. So, how do you use it? You type in a search operation. Here, I think I just put in, can you tell me which eDNA methods, protocol, sample collection approaches, primers, and laboratory procedures are most effective for detecting Eurasian water milfoil in aquatic systems? This is something that I just threw in there. It gives you a very structured response. I don't know how readable that is. So, because I got my PowerPoint in late, what I ended up doing was, from one of the talks from this afternoon, the question about sampling eDNA, how to depth, water mass, spacing, vacuum, decomposition. I threw that in there, and you can see, and I zoomed in this time, and you can see the actual cost, because I've got a range of model selections that we can use in terms of what it costs to run the query. But you can see the first thing it does there is retrieve candidates. It's found the candidates, and in some cases use the markdown, in other cases use the summaries. But we've got the cosine score to go along with each one of these. And then as you go further down, you start to see the structured information. And I just want to draw your attention to what ends up happening. So it started going through the papers, and you can see here it has matched to that PubMed Central record, which you can click on and go to the paper, and the various bits of information for vertical depth and water mass properties. And then if you keep scrolling down, it's a massive report. As you keep scrolling down, vertical depth, depth-related richness and composition, and then going even further, at the very end, you've got this long list of references that were used to generate that response. And it does require more fine-tuning, but I don't really envision this as a chatbot. It's not meant to be a chatbot where you're trying to write your papers, if you will. What I'm hoping is it can be injected intelligence. Perfect example is as part of an eDNA pipeline, where you've got your hit list at the end, where it can take the latest information and contextualize that and say, this should not belong here, because it's got that knowledge. The human review part of that process is where it can encode that information. For example, if you should not expect to find a certain species at a certain depth, that can be surfaced by a system like this that is part of that pipeline. I just need to bring the cost down sufficiently so that it doesn't dramatically increase the cost of each one of those eDNA outputs. But I think this is how LLM automation works. It's not to replace what we're doing. It's to take the massive knowledge base and apply it in a very surgical and thoughtful way. So I'll close on that and say thank you, and maybe there's time for questions. I'm not sure. You have a question? If we have time for questions. Oh, we've got 2 questions. Are we over time, or?
We're a little over time. How much do you need? Okay, maybe one quick question.
Yes, thank you very much, very useful. I want to know, how does it relate with Zotero? Do you have interaction with Zotero? So to input all the papers that we find with your system and then be able to cite them in a document, one. And second, is it similar to LitMap?
To linked map?
Lit map, like literature map.
So literature map would be more citation maps and relationships. This is a pure large language model solution where it's not indexing per se, but ingesting and responding. I can see connections with LitMap because one of the next things that I needed to do as part of the knowledge map is to bring in the citation maps as another knowledge layer. Haven't done that yet, but coming back to your question, I think this is what you're asking. Can you add your own papers to the corpus? Is that the question?
Or all the papers that you find here, can I put them in my Zotero? So then when I write my paper, for example, I can cite those papers.
It should be easy enough to do that, although I'm hoping that's not the end result, but it should be easy enough to do that. One use of this that's obvious is meta-analysis, being able to go through a large number of papers and quickly interrogate and surface things. But what I'm really hoping is that it can become part of pipelines, because I think that's, there's so many eDNA samples that are run. Having that intelligence injected is useful. But I will use this opportunity to address one problem that comes up: open access versus closed. So open access, I can only ingest and serve open access publications. So the solution that I'm thinking is that there is a desktop version of this where the closed papers that you want to add, because most papers now are open access, a lot of them. So you can add that and that becomes indexed as long as it's not served up to everyone else. I don't believe that's breaking copyright law. I should probably check before I make any statements.
Great.
Sorry, maybe I'll hand it back, Bob.
Well, it just, one of the things that I see is we've talked about the need for lists of who's, what primers have worked on what organisms,
Yeah.
and expecting one person to sit and curate a list. Those days are, hopefully over soon.
Yeah.
Right? You can use a dynamic system like that to just query what you're looking for, and it will tell you which primers. Or as you said, we talked a lot about geographic curation of data sets and being able to automate some of this instead of going in manually looking at every little GBIF record and outlier and stuff. This will be a huge boon to our community. We can't wait for further updates, so thank you.
Yeah, I look forward to having it available.
Thanks, Sujeevan.
For our final workshop today, we have our colleagues from the Laboratory Services Division, Agriculture and Food Laboratory here at the University of Guelph. Like you heard earlier, with the DFO lab is an ISO accredited facility. We're going to start with Sujeevan, a colleague who will tell you a bit more about their operation.
Good afternoon, everyone. Thank you very much for sticking with us for the last session of the conference. And we truly appreciate the opportunity to provide a laboratory perspective on how we turn eDNA filters or samples into trustworthy data. So today, representing Lab Services team, we are here to present securing the eDNA workflow, operations meta barcoding, and the qPCR strategies for defensible data. Over the last 3 days, we have seen a rapid growth or expansion of eDNA applications. While the stakes of those eDNA projects continue to rise, analytical labs play a critical role ensure the data are credible. So today we will highlight how we leverage the ISO-based framework to try to get the best data possible. So I will start with the operational baseline under the ISO framework. Then Nicola will discuss the details, how we follow the ISO framework or principles to do the meta barcoding. And finally, Natalia will demonstrate the under the ISO framework, how to do method validation for qPCR assays. So thanks to Francis, who already explained ISO. I guess I can, be quick on that. But basically, ISO is the international quality standard that specifies requirements for laboratory competence, impartiality, and consistent performance. And ISO accreditation matters because it provides global recognition of results, and I would say requires evidence for technical competence, and also promotes risk-based thinking, and proactive prevention. So ultimately, ISO compliance is a mechanism that can ensure laboratory results, not guaranteed, not always steered by human, but the results can stand up in court or under regulatory audit. And when we talk about the ISO framework, we stand on 3 core pillars: structure and the resources, process and the methods, and the quality management. We all know we need to qualify the staff, well-maintained equipment, and a digital capacity. And then, of course, optimize the workflow, validated methods for traceability, etc. But if we only have these 2 components, it's not automatically will happen, will be consistently performed to the quality we need. So there is a component that need a strong quality management. So the quality needs to be managed as well by proficiency testing, document control, audits, and CAPA. It's a corrective and preventive actions. So let's see how we follow those principles and implement those into the eDNA workflow. So we know it's easy to say have qualified staff, well-maintained equipment, but where really sometimes there's a gap, we need to start with the space design and the workflow. So as you know, we are working with very small number of eDNA molecules, and you spend days and so much energy to take the samples in the field. So once in the lab, we don't want to lose any of them. We don't want to mess up any of them. So they're very important. Because the small molecules, we often use PCR amplification technology as a part of the procedure. Unfortunately, all those methods are very susceptible to cross-contamination. So what we do have to start engineer the space at the very first step. So any incompatible activities, they should be very well separated, as you see from the map here. So we have to put the clean rooms on one side of the building and put what we call the dirty rooms, that means post-PCR rooms, on the other side of the building. Maybe only few people even get there. So that well separation, and a separation to the level, you must have controlled airflow, maybe slightly positive versus slightly negative, depends on it's a clean room or it's dirty room. So that type of control, the first thing is the design, make them far apart, as far as you can. And then also the tools we use, they belong to different rooms, so not to switch around. And the lab coats, of course, and we know if you space separated, everybody's lab coats should be different in different rooms. And we had the name tag and the room tag, but everybody was white and it didn't work well. So we ended up with a multicolor system. So anybody wear the burgundy color, you know it's from post-PCR room. You shouldn't be seeing them anywhere near you. So that's how it worked, but you have to engineer this into your process. And then also people, actually we all work hard, especially the research background, we constantly thinking our lab work and want to get the things done faster. Unfortunately, the workflow for people should be unidirectional. So that means you have run the gels in the morning, and if you have no work to do in the post-PCR area, you are done for the day. Unless you can maybe take a shower and have a complete changeover, then start over again. So this sounds extreme, but that's a rigor required to maintain, the contamination-free environment. So of course, all those should be engineered, written in the standard operating procedures in the lab. and then have separate space or designated tools, it's one thing. But one layer of safeguard is not enough. So you need to think about how to protect the samples at every step. So the way we do it, not only in different rooms, but each room have multiple chambers or the PCR workstations to protect your sample whenever you open the tubes, open the samples, open the reagents. So people walking by, there's no wave to your sample on the bench top. So that's a protection you need, another layer. And also, we want to implement automation as much as you can. That's not just for the consistency, reduce the label, also actually minimize the cross-contamination. So there's one actually very critical control point when we do the risk management in the lab for cross-contamination. You need to think where the most dangerous part, which is in the DNA world, it's the PCR products. The potential DNA, of course, as well, but the PCR products are the worst. So just to give you sort of one from the side to the conference, we had actually PCR assays invented in 1980s, right? PCR technology. And we, the lab also do food testing, but the implementation of food testing of the PCR method or technology, there was a 10 years delay. Why? Just the people cannot control the PCR products, especially in the older days when there was no real-time PCR. So running the jails, unfortunately, eDNA, when we provide a prepared library, we still have to run the jails. So that's, I think, the sort of critical control point we must control. So that includes physical control. Means as soon as it's generated, you want to immediately dump in a sealed container, period. And so not let anybody touch it, anywhere exposure. So you have to protect your environment, not only protect your sample. So your environment always want to as clean as possible, And of course, you do all the cleanings you all know, and then how you ensure it's working. So from time to time, you may need to monitor, swab your touching surfaces to see if any targeted DNA can be amplified to be ensured. Of course, you use negative controls and monitor, but on top of that, you can do swabs to test that. So that's sort of prescribed in all the standard operating procedures, how we're supposed to do, how it's supposed to work. So under the ISO framework, we also need to document everything we do. That's a traceability. So the lab services we use called the LIMS, Laboratory Information Management System, to digitally integrate sample from beginning to the end with a unique identifier. And not only the samples, but we also document instrument IDs, method version, reagent lots, expiration dates, calibration records, any measurements about the sample should be fully traceable. And then we have to have a contingency plan for whatever you do. For example, if we claim we want to ensure sample integrity and the reagent integrity, then we must operate a 24/7 environmental monitoring system to monitor every fridge, every freezer, every incubator. We have maybe 50 or 100 in the building, but you have to put probes everywhere to monitor. Then we need to come up with a team of on-call staff, usually managers, directors, and supervisors. So, and even in the middle of the night, if any unit is malfunctioning, an alarm is triggered, and the on-call staff must get up. And sorry, 3 o'clock midnight, you have to move your content to another backup unit. So that's your contingency plan. If you don't have a backup unit, you see where I move, this is not working, but if you have no space to move, that doesn't count. You must have the plan. So redundancy hardware. And by doing those, right, you can sort of make the key point, I would say, it's a continued assurance of the quality by minimizing variability, proactive risk control. For example, with the PCR, you have to predict there's an inhibition or not, and you have to monitor the control the PCR inhibition. So all those components, it's more like I would say it's prepared. Be prepared, not just for what should happen in the lab, which you know, but we have to prepare for what could go wrong. That's always in the back mind. Then you can be proactive to say, okay, what if that happens? So we should be ready for that. So overall, basically there's also, we have people, have instruments, have procedures. That's still not enough. You have to continuously monitor. And the monitor, then you can continuously improve. Monitor actually the PT program, proficiency program, it's one of the mechanisms. Then also audits. The audits have internal audits, have external audits, like we have a QA team regularly conduct the internal audits. So those audits actually help to identify compliance and process gaps, because that helps we improve. And then we also, if any non-conformities or issues are identified, they must be documented. And the root causes have to be investigated by asking 5 why. We call it 5 whys. So why you made a mistake? And it's all because I was not thinking on that day. But why you're not thinking? So basically you have to ask until you figured out truly what's the root cause. But you have to document everything and then come up and implement corrective and preventive actions. That's called a couple. So then the effectiveness of what you come up with have to be verified and monitored over time to see if truly it's working. So that makes it's a sort of the whole process to ensure continuous improvement. So in short, I would say my key message, the quality system, it's a cycle. It's a cycle of monitoring and learning, improving, and confirming what we do today, it's better than yesterday. So with that, I want to conclude my overview with the framework or baseline of the operation. And I will pass the microphone to Nicola, who will walk you through how those principles are put into practice in the metabarcoding. Okay, thank you.
Thanks everyone for sticking with us as well. It's been a real pleasure to hear about all the research that you've been doing. I love working in the lab. It's never a boring day. There's always lots to keep us busy. But after data delivery, I often don't get to see the metadata or the impact or the implication of some of the testing that we do. So it's really interesting to put it into context. So I'm going to walk you through what I do in the lab, which is running the metabarcoding samples as part of a team and delivering the results. So the whole goal of what we're here to do today is capturing biodiversity through metabarcoding, but of course while minimizing as much bias as possible through our laboratory procedures. So we receive numerous types of samples at Lab Services, and therefore whatever projects or taxa that our clients are interested in, whether they be fish, mammals, vertebrates, birds, bats, amphibian species, or where my background actually originally was in soil biodiversity, so things like bacteria and fungi. So those samples can take a wide variety of forms, whether they be water samples that we have to filter in the lab, or for anyone lucky enough to go out into the field who've done the filtering in the field and send us the eDNA filters, we will process those. Things like soils or sediments, as was introduced earlier, whether that be guano for bat species identification from where it originated from, stomach contents of various animals, or as recently started happening, air sampling and plants for any plant pathogens. All those sample types come into our lab, and we have to use our expertise to basically extract the DNA and get the best quality result from that DNA while separating it from a wide variety of sample types that can offer various challenges. And their end goal really is to profile very complex and very diverse groups of organisms. So in brief, what I'm going to present to you is the eDNA analysis high throughput sequencing workflow. So in brief, it's DNA isolation, with the real goal being as much DNA and high DNA recovery as we can get from that sample as possible, but ideally with a low amount of inhibition. Following the DNA isolation, obviously, and it's going to sound very simple, we just do PCR and indexing as part of the library preparation for high throughput sequencing. But we need to have an optimal PCR setup to really maximize the outcome for that data. And that data is generated by high throughput sequencing and then subsequent data analysis, which sounds simple but can actually be quite a lot of the work, as we've had many discussions here about the complexities that can be involved in that. But from the sequencing side, we want high quality sequences at least, which will then generate high quality results. So each sample we have, each target, each project requires attention to the unique nature and any complexities that eDNA workflows encompass, much like the ecosystems that they profile. So how can we maximize DNA but minimize inhibitors? I'll just go over this briefly, as many are probably very familiar with DNA extraction. But for example, when we take an eDNA filter, decisions do need to be made how to separate the DNA that we want from the material that we don't want. And so even in the lysis, and subsequent steps, there are many considerations, decision points that need to be made. How much lysis volume are you going to use? If your sample has a lot of inhibitors, you may want a larger lysis volume to deal with that. But at the same time, that may then mean any species that aren't in high abundance may become more difficult to detect, depending on how much of that lysis is carried through in the extraction procedure. You have chemical and/or mechanical lysis methods to choose from. But as we've seen with like gram-positive and gram-negative bacteria, if you use too harsh of a lysis and extraction, you actually risk shearing your DNA, and that's going to limit your ability to detect certain organisms, even though it may be required to actually get the DNA out of a different subset of organisms. There's not really one size fits all. Are you going to have to do any physical separation? So for some samples, there's so much dirt or material that even after lysis, you're going to have to physically separate that out, which is another manipulation step. There's, with every manipulation, the potential loss of DNA along the process. But are you going to need to do that by either spinning down your raw lysate, running it through a Chi shredder before you have a cleaner lysate to move forward through to capture on a spin column, for example? Even in the elution, there are considerations. Well, ideally, we want to capture anything that's rare or not particularly abundant. So do you want to choose a more concentrated, low-volume elution? But as we've sometimes encountered in the lab, if you want to do multiple assays, multiple markers, multiple replicates, well, if you're eluting in 100 microliters, you may not actually recover that 100 microliters. You may be at risk, especially if you want to use something like 10 microliters per 50 microliter reaction. Well, suddenly you're going to run out of DNA to run all the markers you want, or potentially have more DNA left to go back to maybe a few years later when you want to profile different things. So that's a difficult decision sometimes to be made up front. Okay, I was going to say, I think we've lost everything except the screen that I can see. Well, I'll keep going. For the rest, we don't need the slides. So finally, and especially because we're talking about samples that where the DNA may have inhibition, do we need to do any subsequent purification to the samples? Whether that's a whole separate, running it through a column, or using a bead-based purification or a further kit, there is always the risk that we're going to lose some DNA that's low abundance, but it may be required to get any form of result in the end. If we don't do purification, then if there's inhibition in the sample, you may need to dilute 10x, 50x, 100x. At that point, something that wasn't very abundant might not get detected anyway, and you may be better off choosing a purification method. So take-home message there is samples can vary. Each must be treated consistently, ideally, because you want to make comparisons between your samples. But you also need to ensure the highest quality DNA for downstream use. So sometimes not every sample is going to be the same, and you may need to elute one slightly differently, for example. So I'm going to talk about primer selection. I think we've had a few, we've touched on this topic a lot in this conference, but in primer selection, often we're working with bias. We're trying to minimize that bias, but there's always going to be certain species that are preferentially amplified by certain primer sequences than others. And we had a look at this using an 8 species DNA control from species that we had in our lab. And we targeted it in 2 ways. On the left-hand side, we used a COI metabarcoding primer set. We call it the vertebrate primer set. And we also used that exact same DNA mixture for a mammalian 16S marker. And on the left-hand side, the top left, you'll see that there's fairly equal representation of all the different species. However, not for dog, which is Canis lupus, and mouse, Mus musculus. Those are almost not detected at all. They always end up with very low reads present, but still very low with that particular primer set. If you amplify that DNA with the mammalian 16S, all of a sudden mouse is the top one that is detected, or in terms of its relative abundance. So the primer set we choose is going to offer a different profile, and that's something important to consider. We've seen that as well with fish. We have a 3 species mixture that we've recently been using, and we've used that with both a 12S, commonly used at my fish, and a fish 16S, and they don't give you the exact same relative abundance within that control sample, even though those are amplified at the same time and they were run on the same run. So something important to consider is always what primer set will best capture my tags of interest, because they're all going to pull out different things. There's also specificity to consider. So we do a lot of fish metabarcoding, 12S being a popular one, or one that has become a little bit more established. And what we did as part of a previous project is, it came up I believe yesterday, is some of the fish primer sets will also pick up non-fish targets, which if that's actually something you're interested in is great. But if it isn't, that will actually take up some important reads within your sample. So as part of an experiment, we did DNA spiking, 10% vertebrate DNA, so a mixture of our vertebrate control, in fish DNA using the, on the left-hand side, the MyFish UF, UR, and EF, ER. And the top thing that came out in that control, even though it was only spiked in at 10%, was cattle, followed by a fairly high representation of the other vertebrate species. Whereas a modified fish primer set, which is displayed on the right, the MyFish URFR, those species still come up, the vertebrate species still come up, but to a much lower extent. So depending on what you're interested in, the primer selection is important to consider if fish are your primary target. Now, 10% was a fairly high spiking rate. We also tested this at 1% with very similar results, although at 1% fish are being pulled up more than the vertebrates. So this is important to consider because depending on your sampling timing and what's in the environment, a primer set that worked really well to detect lots of fish in a particular event, if the samples change or what's in the environment change, then all of a sudden they may start picking up other things that weren't expected because the environment is not static. So if that is a concern, always ensuring the appropriate primer specificity, that really just ensures you're getting more data towards your target taxa. So for the PCR amplification, to maximize the outcomes, I feel there's a menu of enzymes that you are able to select from, some of which are high fidelity, others which are maybe more considered standard. There's a number of decisions to be made. Do you go with something that's very degenerate in order to capture as much as possible? That can lead to a lot of non-specific amplification, which is very difficult to deal with later. Or, as we sometimes employ, use a cocktail of primers so that it reduces the degeneracy, but you're still able to get all the taxa that you're interested in. If you do get non-specific amplification, how are we going to handle that? We don't really want any reads going towards that. Options can be gel purification, size selection, or just sequence it and deal with it in the data later. There are pros and cons to each. Also, which enzyme primer compatibility to use, which combination to choose. We have found that primers with inosine bases, although they give you flexibility, they don't work very well with proofreading enzymes. And last, published amplification conditions may not work. We have found that despite following a paper exactly, we were not able to get a recent fish 16S primer set to work, despite extensive molecular biology experience from the team. In the end, we had to try as many other enzymes as we had in the lab until we finally were successful. So in brief, for what we do, library preparation, in terms of trying to streamline the results, there's PCR amplification setup. We often use robotics to try and automate as much as possible. But the keys are we're assessing how has that amplification worked? Is there non-specific amplification? How can we deal with that? Clean up the libraries and do the required quality control in order to have good quality libraries from which we know the fragment size and the concentration. So that would go ahead and pool them. So for pooling, if you have, let's say, a plate of samples, there's various ways to normalize those, whether you do it by equimolar ratio, so we want every sample to have the same concentration. Alternatively, we could normalize by volume, simply pool 5 microliters or whatever volume, and pool them together and then normalize that for your appropriate loading concentration. And the question is always, should samples with more amplification have a higher proportion of data? And that would be captured by normalization by volume versus equimolar ratio. One problem we do encounter, and this is a pro or a con, is the proportion of negative samples, or things with no target amplification, in a library pool will actually have a dilution effect of that library pool before you put it on the sequencer, which basically just reduces your overall data that you're getting for your samples. However, there is a lot of biological meaning to the field negatives that you may be collecting, the laboratory extraction negatives that are monitoring the process, the PCR negatives. You want to get data from those samples, but without reducing the overall data amount that you're getting from the sequencing run. So how to pool those and reduce that impact is something that has to be considered. And so for high throughput sequencing, the best is to use multiple markers to provide the base diversity that you need. So combining multiple targets in one run is usually the best way to do that, especially now that sequencing costs are getting lower. You're able to put more and more markers on a single run. So instead of having to select one marker for one type of taxa of interest, you can actually go and put more than one on now. And so that will provide that base diversity. But one thing to consider is the sequencing technology. And that is, some technologies will perform better when you have, let's say, for example, a 2x300 read length. maybe not all of your markers require that. You'll end up with adaptive read-through, which I'm sure some of you may have encountered before. Illumina can handle that to a certain extent, but one of the other systems, the Aviti, it is a great instrument. It has very high data quality. It's less expensive and generates a lot of data, but it performs sequencing in a different way than Illumina. It forms a polyny, which means when the sequencing occurs, it reads through your fragment. It can actually read through into the adapter and read through into another version of your fragment, which means when the data comes off, it can be extra challenging to handle that. And so for the data processing, if there is non-specific amplification or primer dimers or things like that, as your depth increases, we're generating more and more data as the price comes down. The computational analytical burden of any non-specific products is a challenge. So I'm going to pick on my fish a little bit again. It not only amplifies vertebrates, but and can have some primer dimers, but it often amplifies bacteria if there happens to be a lot of bacteria in your sample. And that product is unfortunately larger than the target, which means we can't purify it out using standard size selection methods. So when it comes off in the data, there are various ways, if you're lucky, that you can handle that in order to really narrow down and get the best outcomes from the data analysis pipelines, which wants the cleanest data as possible. The dimers can be handled by a primer trim using cut adapt and establishing a minimum length. That usually gets rid of anything short. But in the denoising step, the key would be to try and truncate your sequence so that anything larger than it is immediately excluded when you have to overlap. That will generate the best quality ASVs and therefore the best quality taxonomic assignments. Now I must caution that variation in amplification products between samples, runs, or projects, you may have non-specific amplification in one project but not in another, means you have to pay attention and modify these parameters when necessary. And last but not least, the databases. This can be a huge source of variation. I'm just going to leave you guys with, for this particular assay, it was a targeted assay towards beverage spoilage organisms, and depending on which database we used, it was the exact same data. It either detected both or only got detection in one database versus the other. I'm oversimplifying, but with the time, and there's definitely not scope in this project to talk about this in full, but yeah, that is a discussion for another time. So thank you, and I'm going to pass it to my colleague Natalie to talk to you about qPCR.
Thank you, Nicola, and thank you everyone for amazing conference. It's been an enormous pleasure to see old familiar faces, meet new people, and exchange ideas during the 3 days of the conference. I'm going to talk about validation framework for qPCR assays today. We've heard enough from previous talks, especially today, that we work on definitions, we work on standards, we discussed what is truly positive, what is truly negative. And in a lab services environment, being in ISO certified lab, we have to create validation framework for assays. Because so many publications out there, some of them work, and then let's say when we get requests to establish particular assay, we have to go through a process of selection of assays first, and then we need to apply what we do in the context of IS certified workflow. That's a good example of references for assays which I will be presenting today. And why do we need that validation framework in our IS certified environment? First of all, we want to deliver precise and accurate results. We want to do it in a cost-efficient way and within time constraints, because we really need to deliver those assays to clients fast. We cannot take months and years for these validations. So we had to come up with some kind of streamlined workflow. First of all, checking for human errors, because some of the publications might be publishing wrong primer sequences. Some of those primers and assays published might not be specific enough they might not be validated to the quality standards which we need. They might have reduced performance, and they might not be scalable for testing in our environment, and they might not be compatible with our standards. So first of all, while talking about scalability in the testing lab, we need to make sure that assay which we're pulling in the lab environment is compatible with our cycling conditions, because I don't want to have 10 assays, each with annealing temperature. We do have some assays which have enough variability, but I want to ensure that we can streamline at least one of the workflows and to make sure it's really easy to run to begin with. We want to make sure that there is no influence of the qPCR instruments which we use, because instruments might be different between different facilities, and of course there is our master mix, because people use different master mixes, combination of primers and probes, and compatibility with our internal control, which we use across all different assays which we run, not only for qPCRs. We have many different assays. We use the single internal control for many, many different assays, and we want to make sure that the new assay which we introduce is fully compatible with that control. So we developed validation pipeline, first starting with literature search and doing all basics checks. Then we test assay for functionality, specificity, efficiency, and linearity. Then we establish limit of detection, and then we challenge that limit of detection by placing and diluting our G-block in environmental DNA metrics to ensure that LOD still stands. Because if assay is weak, you will lose your LOD while moving to environmental sample. And finally, we ask ourselves, what if something goes a little bit wrong? What if we use different cycling instrument? What would be the difference between the operators? And what if our reagent load is different and slightly different volume of that reagent was used? And finally, if we encounter positive in the assay which was newly introduced to the lab, we do Sanger sequencing. So those are our metrics. Some of them we didn't go for extreme detection limits. We wanted to be consistent, at least at 10 copies DNA minimum, but we also evaluate performance of assay validation by calculating standard deviations and relative standard deviations across replicates as we validate the assay. A few remarks about the probe choice. You need to ensure for environmental DNA that your signal to noise ratio is good and that your sigmoid curves are steep. You need to check for the length for the probes, and you need to consider cost-efficient labels, but sometimes you want to go more expensive if you want your background to be clean. You have to consider manufacturer of your probes. There are so many different providers right now. You need to consider dye compatibility with your particular qPCR instrument which you're using, because some of them might not be suitable to run certain dyes. And there are several choices. One of the common choices is just your dye and quencher probe. You can have fluorescent, non-fluorescent quenchers, but some of the probes which are really good for qPCR assays is double quenched probes, because they enable that lower background and allow higher signal-to-noise ratio. So you can also use, if you have really short probe and you have short probe constraint, you can use linked nucleic acid probes, or you can also MGB probes with minor probe binders, because those probes allow short ratios. I'm not going to go to explain this more, because this community is really aware of the types of the probes, but those are choices which can be useful for DNA. Don't go extensive specificity testing, because usually a validated published assays already established that. We also can check it through alignments, for blast searches, and that's done at the initial stage. But we want to ensure that qPCR assay doesn't amplify some common species. So to do this, we have several panels. We do have a fish panel, we do have invertebrate panel, we do have some common species, commonly occurring, let's say in the rivers. You can get cow DNA, pig DNA, horse DNA, dog, cat DNA, and of course human DNA. So by running assays through those 3 panels, we ensure that the assay would not pick up common contaminants. This is an overview of assays validated today in the lab. I would say that most of them, except for the spotted turtle assay, which was just below our expected threshold for efficiency, it was slightly lower, but this is the longest assay. They all successfully passed, and some of them exceeded our expected LOD thresholds. Those are examples of standard curves for 2 latest validated assays, which are sea lamprey and Jefferson salamander. And they're quite good in terms of repeatability and reproducibility, even though you can see that some assays would not be, they would have wider span between replicates, while some other will produce really tight replicates. We also challenge assay by operator. At the time when we establish an idea, we have certain data points repeated by different operator to ensure that our repeatability withstands our expected criteria. And of course, there was a lot said about inhibition control. So we do use internal control, which is plasmid. We dilute it to the point when it's not really competing with our qPCR assay, and we also use lower probes and primer concentration for internal control to make sure it doesn't draw resources. And we test, as I mentioned before, we test our target G block at the LOD and level above to ensure that LOD stays at that limit. You can go below LOD and qPCR for environmental DNA, but in this case, replicates would matter. This is example of 12 replicates below the established LOD for Jefferson salamander assay. And you can see that only 2 replicates worked for 1 copy of estimated G block dilution. Of course, there is measurement uncertainty, and in IC certified environment, we mitigate this as much as possible by integrating our QC measures, traceability of reagents, calibrations of instruments, use of internal control. There's still some uncertainties, but those uncertainties would be controlled by collecting metrics during the validation. So at the end, those uncertainties would be minimized in an IC certified environment by continuous improvement and by constant monitoring of results. This is example of heterogeneous testing. Sometimes you can have panel of qPCR instruments and you want to make sure that they are calibrated, but you want to make sure that they still perform the same on the assay. Your reagent load can differ slightly from one to another, or storage conditions of this reagent load was different. What we typically do while validating qPCR assays, we challenge our master mix by keeping it at room temperature overnight in a closed dark environment, and then we test it next day. We also test reagent volume variability by adding a bit less or more with the master mix. And this is example where you see virtually very little standard deviation and very little relative standard deviation as example of validation Jefferson salamander assay. For the field detections, we do use Sanger sequencing, and sigmoid curve was already mentioned today, and this would be a good example when you have a lot of that some background control, but it's not sigmoid curve. While those three would be a real one, and those are detections. This is example from Blanding's turtle assay. So at the end, those are the references which I mentioned in the beginning for all those validated assays. For the most of them, we didn't have developed anything else except for one probe. And to conclude, I would like to emphasize once more that there are so many assays out there. We need to create better libraries of those assays, making them accessible to other researchers so they can be widely used and the validation process can be streamlined. And I got frequent question during that course of the conference about location of the lab services. We're actually on campus and we're facing Stone Road. This is our beautiful building facing the Stone Road while you walk through the university towards the main campus. You usually bypass it, sometimes not knowing that it's there, unless you're going to our sample reception. So here we are, facing Stone Road, we're on campus, and we're accepting many, many different samples, including microbiology, eDNA, milk testing, toxicology, name a few more. Number of samples and number of methods performed at lab services truly blows my mind. Thank you.
Thank you very much. I think it's been a nice tour de force to help, particularly for the students, to recognize this is not for the faint of heart. If you're going to be generating, legitimate, defensible results, there's an awful lot to think about, and there are experts that you can draw on to help support you in some of these projects. I know for my lab, for things that do have regulatory aspects or potentially litigious aspects to them, we run the samples through laboratory services and then do data interpretation through the students and postdocs in the lab. Other things we generate in our own lab, in an academic lab, where you may not have quite so much riding on it. But it's nice to see DFO has established their own lab that is moving in this direction, that is ISO certified. I think this is something that you really need to think about as you begin to operate in this space, is thinking about, as we heard this morning, what's your question? What is your tolerance for risk? And that will help you kind of decide the level of service provision you need. With that, I know we're running a little overtime. Some of you have long commutes or flights to catch. I want to just wrap up by quickly announcing the two students who won the oral presentation prizes from our keynote speaker judges. Christine McPherson, still in the audience? Congratulations. Thank you for your contribution to the conference. And Sanchi Singh. She had to leave? Okay. Well, we'll email her and notify her. All right, that concludes Act 3 of PISCeS. Thank you so much for your attendance, and I hope you found the meeting worthwhile.
Thanks, Bob.
Oh, and I think I would be remiss in not thanking all of the volunteers, Kate, Kevin, Helen Vanos, and all of the student volunteers. It takes 1,000 details to be attended to pull off a meeting for 100 people like this. So without them, we couldn't do this. Thank you.